order. Okay, so presentation will be held on the week 14, waktu lab. Okay, week 14 sepatutnya kita dah habis kelas amali lah. Kan? Kita boleh buat. So we have, yep.
Tak tahu kenapa tapi tiba-tiba doktor punya suara macam bergema.
Alamak. Okay, saya baru pakai earphone. So dah okey ke belum?
Okey doktor.
Okay, clear. Alright. Okay. So I hope you all semua dah tahu about your SCL. Kita akan buat on week 14 waktu lab class. Week 14 sepatutnya kita dah habiskan amali. So we have 3 hours. I hope you can finish all berapa group? Maybe 9 to 10 groups, okay? In that 3 hours. Okay? So we agree, me and Dr. Nina agree to give you freedom to choose your own groups, but totally different group daripada lab. Okay? Kami tak nak ahli yang sama. Alright, if you can mix around, try to work with different people, it would be good. Okay? Alright, let me share screen. Nampak ke screen saya? You all nampak apa?
Nampak doktor, nampak slide.
Nampak slides? Okay, alright. Okay, so we are done with DNA metabolism. So basically on DNA metabolism, we have discussed on the replication. Okay, if you still remember the central dogma is a very important system. Okay, it starts with replication, transcription, translation. All right, so on DNA metabolism, we have discussed on replication. So this topic, okay, this new topic on RNA metabolism, we move to transcription. So what happens during the transcription process? What are the principle behind it? What are the mechanism involved? So how does the transcription process occurs in both prokaryote and eukaryote system? Okay. Okay, so what is transcription? So it's a process of synthesizing RNA molecules from DNA. Okay, in simple words, in simple sentence, we want to synthesize RNA from DNA. Okay, so kita dah ada DNA dari yang kita dah replicate. Okay, we make a copy, thousand millions of DNA. So now we want to transcribe it into RNA. Okay, why we need to transcribe it? Because we already make a copy of all the genetic information. So now we want to transfer all the genetic information from DNA to RNA. Okay, so that's what it means by transcription. Okay. All right, so these are the 4 major types of RNA molecules. We have mRNA, we have tRNA, we have ribosomal RNA, and also small nuclear RNA. So what is messenger RNA? Okay, so these are the nucleotide sequence that direct the protein synthesis. So it encodes the amino acid sequence of polypeptide. So it helps in the as a messenger for the protein synthesis. Okay, so what is transfer RNA? So transfer RNA is a small compact molecules that deliver the amino acids to the ribosomes. Okay, it brings amino acids to the ribosome during the translation. Okay, and ribosomal RNA. Okay, we know what is ribosome. Okay, so in RNA molecules, we have this ribosomal RNA, and it constitutes 2/3 of the ribosome. So 2/3 of the ribosome is ribosomal RNA. Okay, RNA. So it combines with proteins to form a ribosome. Okay, that act as a catalyst for the translation. Okay, and for the small nuclear RNA, it combines with protein to form complexes used in eukaryotic RNA processing. Okay, so these are the general features. What is transcription process? Okay, what is transcription process? So transcription, or other terms, okay, known as a gene expression, okay, is regulated by gene regulatory element associated with each gene. Okay, so it is a control process. Okay, it has the regulatory element, so it has a specific gene that we want to transcribe, and it has all the regulatory elements that control the transcription process. Okay, and one thing that you need to know is by having this topic, RNA metabolism, you should be able to differentiate, okay, between RNA metabolism and also DNA metabolism. What is different between transcription and also replication? Okay, so for RNA transcription, RNA is transcribed from 5' to 3'. Okay, which we know that template DNA strand is read from 3' to 5'. Okay, and the polymerization of RNA somehow is basically similar to DNA synthesis, similar to the replication, except for the precursors for RNA synthesis NTPs. Okay, nucleotide triphosphate, not DNTPs, because in DNA synthesis it involves deoxynucleotide triphosphate. In transcription, there is no primer needed, okay, to initiate synthesis. Okay, kita tahu kan dalam replication we need primer. We have primase to synthesize primer, and this thing I'm sure all of you know that in transcription in RNA synthesis, uracil is inserted instead of thymine. Okay, there is no thymine present dalam RNA. Okay, so these are the general features for the transcription process. So what are the similarities between replication and transcription? Okay, both processes use DNA as the template. Okay, semua perlu ada template. Okay, they need template. Okay, so what are the templates? So ia adalah DNA sequence. So DNA act as a template for the replication, for the transcription process. Okay, and another similarities between replication and transcriptions is phosphodiester bonds. Okay, these are the linkages that form in both replication and transcription. Okay, in the RNA, in the transcription and also replication. Okay, and both synthesis directions are from prime. Okay. Okay, so these are the differences. Okay, we have the similarities and we have the differences between replication and transcription. Okay, so template for replication, we know it's all started with double helix DNA. So it's a double strand DNA, but for the transcription, the template would be the single strand DNA. Okay, and substrate for replication is the NTP, but for transcription is NTP. Replication require primer, but not in transcription process. In terms of enzyme, replication require DNA polymerase, but for the transcription process, it has their own RNA polymerase. And the product would be, for the replication, is double-stranded DNA, but for transcription is a single-stranded RNA. Okay, we can see a difference in terms of base pairing between in replication we have this A, T, and GC, but for transcription, obviously T will be replaced with uracil. Okay, and we have this T and C. Okay, all right. Okay, let's look on the process itself that involved in the that occurs during the transcription process. Okay, so the transcription is divided into 3 steps for both prokaryotes and eukaryotes. Okay, dua-dua system. Okay, prokaryotes and eukaryotes, we can divide it, the process into 3 steps: initiation, elimination, and termination. I think I already mentioned about these 3 important steps. Okay, and these important steps in the application, we can understand the process when you conduct PCR. Okay, polymerase chain reaction. Okay, polymerase chain reaction also involves these 3 important steps: initiation, elimination, and termination. Okay. The process of elongation is highly conserved between prokaryotic and eukaryotes, but initiation and termination are somewhat different. Okay, it has it's not similar in for initiation and termination between eukaryotes and prokaryotes. Okay. All right. Okay. Okay. In the replication, okay, we can replicate the whole genome of DNA. Kalau genome E. coli, it consists of the size of E. coli, for example, of that bacteria is 300 kilodalton per after. We still can replicate the whole genome. Okay, kita boleh buat copy. In fact, the whole genome. But for the transcription, normally only small portion, only the specific gene, okay, that required, that needed physiologically, or it required because of the environmental changes, is going to be transcribed. Okay. Even though kita buat copy the whole genome, but only specific gene, only small portion of genome that required for the transcription process that we need to translate, that we need to produce a protein, is going to be transcribed before it can be translated. Okay, tak semua. Contoh, kita copy the whole genome, the whole human genome, tapi we want to get protein of blue eyes, for example. So kita hanya akan transcribe the specific sequence that code for blue eyes, for example. Yang you nak clone contoh dalam haiwan ke, kan? Maybe you want to do some hybridization between blue orchid with yellow orchid. So you want to hybridize that the 2 colors. So you are going to transcribe the specific gene that code for blue color for that orchid, and you want to clone it into the yellow orchid so that you can get a very nice blue yellowish color of orchid. So only specific gene, only small portion of genome that's going to be transcribed based on the requirement, on the physiological need and environmental changes. Okay, and DNA regions that can be transcribed into RNA are called structural genes. Okay, structural genes is whatever genes that whatever sequence, whatever genes that you want to transcribe, that need to be translated into proteins, are called structural genes. Okay, ada soalan? Okay.
Yep, contoh maksudnya kita copy whole gene, And then dekat dalam gene tu ada exon dengan intron tu ke yang dia maksudkan tak ambil the whole sequence as in untuk transcribe.
Okay, exon and intron is a molecule that involved in the process. Okay, yang dimaksudkan, okay, is it do you mean this statement only small portion of genome is transcribed? Okay, contoh macam ni.
Maksudnya
Alright.
dia tak
Okay. I understand your question. Okay. Let's say we have the whole human genome sequence. Okay, we can make a copy, we can replicate the whole genome. Contoh kalau kita berapa size genome, human genome? Siapa tahu? Human Genome Project, berapa size human genome? It's not fully sequence lagi. The project is not completed yet. Okay, but they are working on it. Okay, apa tahu the human genome size? Okay, we can replicate the whole genome, but apa yang ditranscribe is whatever structural genes that required based on physiological need, based on environmental changes, in response to development requirement. Contoh, okay, baby, okay, during the baby's growth, kan dia ada dia punya face, okay, from baby, daripada tak ada rambut, dia ada rambut, betul? So at that, during the growth phase, okay, so that our the body system, dia dah tahu dah bila masa dia nak transcribe protein that produce rambut. Contoh kita punya keratin, kan? Keratin is a protein that present dalam kita punya rambut, kan? Okay, so our body knows, okay, bila masa, okay, in which growth phase of the baby, specific the structural genes that code for keratin need to be transcribed and translate. Even though kita ada the whole human genome, tapi certain phase, okay, mungkin dekat umur 1 tahun, 2 tahun ke, okay, it start to be translated. So that specific keratin, specific gene that code for keratin needs to be transcribed and translated. Contoh, kalau lah kita ada mutation, radiation ke apa ke, kan, and one of kita punya protein that code for, contoh, protein kita yang code for kita punya, contoh apa contoh kita punya
insulin.
Insulin, might be, okay, boleh juga. Mungkin kita our body system dah tak boleh nak produce insulin, for example, kan. Mungkin either kita boleh ambil external insulin, okay, from injection, or mungkin we can take maybe our body system will produce, saya tak tahu the exact mechanism, maybe our body system can produce other proteins that can tolerate, okay, bila kita punya badan dah tak ada insulin, contoh. So our body system knows, okay, dia ada dia punya own, sebenarnya kita punya body system ni the whole system is very kita tak terfikir pun, kan. So it's trigger, it knows what kind of protein, what kind of what our body needs. Okay, so it only specific, apa saja spesifik yang diperlukan, itulah saja yang akan di-transcribe and translate. You imagine kalau the whole, okay, dalam the whole genome, human genome tu, we have I think million. Okay, million of genes. Okay, itu pun banyak lagi yang belum discover. Okay, can you imagine kalau semua benda is translated, is transcribed and translated, what happen? Okay, kalau kita punya growth hormone, if you do growth hormone, kalau we have excessive of growth hormone that be translated into protein, nanti kita akan ada ketinggian yang terlalu lebih. Contoh, okay, we have rambut kita terlalu panjang, kalau excess, if the translation of the protein is not controlled, our body system akan haywire. Okay, it's not it needs to be transcribed and translate based on the physiological need. Itu maksudnya. Okay, semua boleh ditranslate. Okay, apa saja yang diperlukan, dia akan trigger, dia akan bagi signal, and that specific sequence, that code for that gene is going to be transcribed and translate into protein. Okay, apa saja contoh kita punya kulit, apa protein dia yang you tahu? Contoh rambut keratin, kita punya nail pun keratin. What else?
Collagen. Collagen.
Okay. Collagen. Okay. So that is protein. Okay. So let's say kalau contoh if our system don't have enough collagen, so dia akan trigger, dia akan bagi signal, so the body needs to start to synthesize the specific gene that codes for collagen. So collagen lah yang disebut bila kita dah tak ada enough collagen. So it based on physiological need, it based on apa saja yang badan kita perlukan. Kalau ada dia affect due to environmental changes ke apa, so dia akan bagi signal to the body system untuk start transcribe and translate only specific gene that required. Okay, tak semua boleh translate and transcribe. Kalau tak ada, if the translation and translation processes is not controlled, is not regulated, I can't imagine. Contohnya kalau kita punya growth ke, kita punya nail, kalau kita punya keratin is not control, asyik translate, just what happen? Kita punya body system. Okay, boleh faham tak?
Faham Doktor?
Okay, alright. So now let's look on the template. Okay, template for the transcription process. So you dah tahu kan, template untuk replication is a double-stranded DNA. Double-stranded DNA that unwind, that being broken down by helicase. Itu dalam replication. So template in the RNA, in the transcription process, is still DNA. Still, the main template adalah DNA. Okay. So the template strand is the strand from which the RNA is actually transcribed. So ini it also also known as an antisense strand. Kita ada dua term, antisense and also sense strand. Okay. All right. So the DNA strand that serve as a template during transcription is known as an antisense strand. Antisense yang act as a template untuk RNA transcription, antisense. Okay. So kalau untuk sense strand is the coding strand. Okay. The coding strand is the strand whose base sequence specifies the amino acid sequence of the encoded protein. Okay, is a sequence that complementary to that of the RNA. So ini adalah sense strand. Okay, yang complement to antisense dipanggil sense. That's it. Tapi yang act as a template adalah antisense. Okay. Okay, so what is asymmetric transcription? So what is asymmetric transcription? All right. Okay. Okay, only the template strand is used for the transcription. Okay, but the coding strand is not. Okay, hanya template strand. Okay, so in terms of the direction, okay, of the transcription is it occurs as in opposite. Okay, and this feature referred to as the asymmetric transcription because transcription hanya di-initiate pada template strand. So bila berlakunya transcription, the direction is pada different strands kan, dia berlaku opposite. So this is known as an asymmetric transcription. Okay. Okay, so let's look on RNA polymerase. So these are the enzymes that responsible untuk transcription. So what is RNA polymerase? Okay, so this is known as a DNA dependent RNA polymerase. Okay, maksudnya dia adalah enzyme that because dia menggunakan template DNA template kan, so this is known as a DNA dependent RNA polymerase. Okay, so it has two in both prokaryotic and eukaryotic system. Okay, dua-dua present RNA polymerase, but the difference is eukaryotic system. We know that eukaryotic system is much complex system. So eukaryotic system has 3 kinds of RNA. Ada certain books that mention dia ada 4, 5 RNA polymerase. Okay. Right, and it is responsible for transcription of different RNAs. Okay, so basically all cells contain RNA polymerase. Kira lah prokaryotik ke eukaryotik semua ada RNA polymerase. Okay, let's look at the structure of RNA polymerase. Okay, so RNA polymerase is a holoenzyme. Okay, what does it mean by holoenzyme? Siapa ingat holoenzyme?
Dia tak activated tanpa cofactor.
Alright, correct. Okay, so you need to know what it means by holoenzyme. So this is holoenzyme. And in E. coli, it consists of 5 different subunits. Okay, it has 2 identical alpha subunits. It has beta subunits, beta prime subunits, omega, and also sigma. Okay, so these are the and these 5 different subunits, okay, made up a holoenzyme of RNA polymerase. Okay, so what is the role of each subunits in RNA polymerase? Okay, so this alpha subunit, okay, it determine the DNA that to be transcribed. Dia sebagai macam it identify, okay, identify DNA yang akan undergo transcription. Okay, both alpha subunit, 2 identical subunit alpha. And you have this subunit beta that catalyze the polymerization. Subunit beta prime that it is responsible in binding and or opening the DNA template, and sigma factor that recognize the promoter for synthesis initiation. Okay. All right. One thing that you need to know, once RNA, kita tengok dekat gambar ni. Okay. Sekejap lagi ada gambar. Alright. So RNA polymerase also contains 2 zinc molecules. Kalau if you have the opportunity untuk cari X-ray structure ke gambar enzyme structure of RNA polymerase, it contains 2 zinc molecules. But there are some books that stated that it contains magnesium, iron, and also zinc. Okay, zinc ions. Okay, so dalam ni saya letak 2 zinc molecules lah. Alright, and the core RNA polymerase associated with a specific protein factor, dipanggil sebagai sigma factor. Okay, certain books dia tulis sigma factor 70 dekat situ. So the sigma factor is one of the subunit that plays a very important role in recognizing and binding to the promoter region. Because of the sigma factor, dia akan initiate the transcription process. Alright. Alright, so this is, okay, with these 5 subunits, okay, if you look at this figure, okay, all these 5 subunits made up holoenzyme. Okay, dia akan jadikan RNA polymerase as a holoenzyme dengan segala cofactor. Okay, but once RNA synthesis is initiated, okay, once RNA synthesis has been initiated, the sigma factor dissociates. Okay, the sigma factor dissociates. Okay, from the core enzyme. So these are the core enzymes, okay, of 2 alpha subunit, beta and beta prime. Now it's known as a core subunit, okay. So the sigma factor dissociates once RNA synthesis has been initiated, okay. And sebenarnya RNA polymerase, especially for bacteria, is a very, if you look at the molecular mass, okay, dia punya, it has a very large molecular mass, so it can be clearly visible dalam bawah micrograph, so that if you have time, go try to search, okay, for the structure of E. coli RNA polymerase, okay. So in medical line, okay, this is one of the therapeutic drug, okay, rifampicin, okay, this is a drug that has been used to inhibit the RNA synthesis, okay, and the mechanism of this drug is it binds specifically to the beta subunit of RNA polymerase in the treatment of tuberculosis. Okay, so that's it tells you how important to understand the mechanism involved, okay, in whatever pathway, whatever disease, okay, because it helps if we understand the mechanism, if we understand what happened, if we know what are the enzymes, if we know the direction, so that they can come out, they can design a therapeutic drug that can inhibit the synthesis. Okay, apa maksud dia therapeutic drug? How do you imagine the structure of rifampicin? Okay, contohlah. Okay, let's say it will design a drug that has a similar structure with whatever enzymes that bind to the beta subunit. So that once this rifampicin binds to the beta subunit, so it automatically inhibit, because the next process cannot occur, because rifampicin is only a therapeutic drug. Okay, so that's the importance of understanding mechanism, understanding pathway of whatever process occurs in our body system. Contoh macam kalau you involve dalam cancer research, if you involve in diabetic, cancer, high blood pressure, whatever disease, contoh now COVID kan, so they need to understand the mechanism behind that, okay, apa enzyme, apa molecule, apa substrate, so that they can come out with alternative plan, okay, how to inhibit, how to stop, okay, the process. Do they need to design a new drug to inhibit they need do they need to come up with alternative pathway for the system? Okay, so the key point is to understand the mechanism. Okay, what happened during the proliferation of cancer cells, for example. What are the enzymes involved? Okay, so what happened during the proliferation? So does the mechanism involve outer factor? So all that discussions will help in understanding the mechanism in order to come up with alternative plan untuk overcome disease. Okay. All right. Okay. So just now we look at the structure of RNA polymerase dalam prokaryote system. Okay. We know that in eukaryotes, RNA polymerase, there are just many types of RNA polymerase. Okay, in eukaryotes, okay, there are three different types of RNA polymerase. Okay, that differ in the RNA state synthesize. Okay, we have RNA polymerase 1 that located in the nucleoli. Okay, that synthesize the precursors of most ribosomal RNAs. Okay, we have RNA polymerase 2 that occurs in the nucleoplasm. Okay, and RNA polymerase 2 that synthesize the mRNA precursor. Okay, for RNA polymerase 3, okay, also occurs in the nucleoplasm. Okay, and it synthesizes the precursors of 5S rRNA, tRNAs, and a variant of small nuclear RNA. Okay, and I just add in one important point. You need to know that this is another inhibitor for RNA polymerase, which is called as amanitin. Okay, so lupa lah specific application for amanitin. Okay, we know that it's an inhibitor, but yeah, memang lupa. Okay, alright. Ada soalan? Before we move to E. coli. Okay. Okay. We have discussed on the template on the RNA polymerase for both prokaryotes and eukaryotes. Okay, kita dah tahu structure. Okay, you understand the structure. Okay, what is holoenzyme? So what happened to this holoenzyme once RNA synthesis has been initiated? Okay, so now let's move back to the transcription process dalam prokaryotic system. Okay, dalam prokaryotic system. Okay, in eukaryotes. Okay, most of the protein coding genes are transcribed individually. Kita tahu kan dalam eukaryotes it has 3 different types of RNA polymerase. Okay, so it's going to be transcribed individually. Okay. However, in prokaryotes genomes, okay, genes are frequently arranged in tandem, okay, along a single strand so that they can be transcribed together. Okay, prokaryote and eukaryotes. Okay, so in prokaryote, so this each transcriptable region which is arranged in tandem is called operon. Okay, operon. I'm sure you biasa dengar kan operon E. coli. Biasa dengar? E. coli like operon. Pernah dengar, pernah belajar kan? I'm sure pernah belajar ke? Matrix ke? Saya ingat lupa lah. Tak tahu you all punya
Lack of
lack operon kan. Okay, so that's the system for prokaryote. Okay, so one operon includes several structural genes. Okay, and upstream regulatory sequences or regulatory region. Okay, so in prokaryote system, so dia punya genes are arranged. Okay, genes are arranged in tandem. Okay, dalam satu operon dia ada several structural genes. Okay, structural genes is the one that's going to be transcribed. Okay, dia ada at least 3, 4. Okay, and also in like operon it has a promoter. So promoter is very, very important. Okay, so promoter is in a sequence that RNA polymerase can bind. So basically RNA polymerase they can identify to start the RNA synthesis. You need to identify the promoter. Okay, kalau contoh dalam replication kita ada origins kan, kat mana dia bermulanya replication, so they have origins. But in transcription dalam E. coli system, they have promoter. So this is the key point. So this is where RNA polymerase need they have to identify kat mana promoter. So and this is also the key point for the control of the transcription process. Okay. So this is the initiation sites. Okay, promoter is the initiation sites that are recognized by the corresponding sigma factor. Okay, promoter jugalah yang di-identify oleh RNA sigma factor of RNA polymerase. Okay, ingatkan RNA polymerase structure. So we have that sigma factor. So the sigma factor that identify promoter. Dia akan identify the sequence of the promoter. Okay. Alright, okay lagi semua? Okay, you all letak apa? Tulis dekat notes ni kadang-kadang saya tak baca tau. We transcribe specific genes sahaja. Yes. Specific genes that required by our body system. Bukan specific genes sahaja. Apa sahaja yang diperlukan oleh kita punya body system. Okay. Alright. Okay, so these are the promoter. Okay, these are the prokaryotic promoter. Okay, we have this transcriptional start site. We have this. Okay, this is the coding strand. Okay, this is the template strand. Okay, so we have this promoter. Kita ada this minus 35 sequence and also minus 10 sequence. Okay, so okay, if you look at this figure, the sequence for this okay, kalau this minus 35 sequence also known as the upstream of the promoter. Okay, upstream of the promoter. Okay, upstream of the promoter. Okay, and these are the consensus sequence. Maknanya dia conservedly has been conserved. Okay, this is a conserved sequence for minus 35 and also minus 10. So kalau you tengok minus 10 ni, dia also known as the Pipno box. Okay, so it's a stretch of 6 nucleotides TATAAT. So in whatever promoter, dekat this minus 10 sequence, so these are the 6 nucleotides. Okay, dekat minus 10. Okay, dia dah conserve dah. Okay. Same goes dekat minus 35 ni, these are the sequence, okay, TTG, ACA. So this is the recognition site and the binding site of RNA polymerase. Okay, so The sigma factor of RNA polymerase akan identify dekat sini. Okay, akan identify the promoter at this minus 35 region. Okay, it will identify, they need to identify this sequence, TTGACA. Okay, for RNA polymerase to bind pada promoter, dia kena identify the sequence. Okay, and at the minus 10 region, okay, so this is the region which stable complex of DNA polymerase and DNA and RNA polymerase is formed. So this is, so the minus 10 region is also known as a Pribno box, okay, because of this sequence has been identified oleh David Pribno, okay, one of the scientists who described this in 1975 code, okay, 1975. Okay, it is, okay, so these are highly conserved sequence, okay. Okay, this minus 35 region also known as the upstream, okay, upstream region, upstream sequence, okay. And the other similar sequence juga lah, TTGACA, di conserve juga, okay. Alright. So this is another presentation how the promoter is being presented. Okay, macam mana dia presented selain macam ni. Okay, dia akan ada minus 35 box, minus 10 region, okay, minus 10 box. So these are the consensus sequence of minus 35 region and minus 10 region. Okay. Apa pula minus 1? Yep, minus 1 and plus 1. Yep.
Nak tanya yang Pak Doktor cakap baik minus 35 dengan minus 10 sequence ni recognition and binding site for RNA polymerase kan? So maksudnya RNA polymerase ada dua ke?
Bukan. Okay. Okay, for the initiation, for the start of the RNA synthesis, okay, RNA polymerase, okay, specifically the sigma factor tu, dia akan identify this minus 35 region. Dia ada recognition site, so so the sigma factor of RNA polymerase needs to identify, dia perlu cari this sequence, TTGACA. Okay, because this is the binding site. Okay, once it binds dekat sini, so dekat minus 10 region, dia ada this consensus sequence TATAAT. So it helps to stabilize the binding between DNA and RNA polymerase. Okay, dia punya binding site, dia punya recognition site adalah dekat minus 35 region. But this Pribno box ni, minus 10 region ni, it helps to stable the complex. Okay, because nanti akan melakukannya transcription dekat sini. So this is the transcription start site. Kat sini adalah recognition and also binding site. Okay, once your RNA polymerase binds to this minus 35 region, so dekat sini we have this Pribno box, this 6 nucleotide sequence ni, it helps to stabilize the complex between DNA and also RNA polymerase. Okay, so that later barulah transcription boleh berlaku. Okay, so kat sini you boleh tengok juga ada plus 1 and minus 1. Okay, so what is plus 1 and also minus 1? Okay, plus 1 or minus 1 ni adalah dia macam is one nucleotide that it's normally this is the initiation nucleotide lah. Okay, plus 1 is the initiation nucleotide. Okay, it's always either A or G. Dia panggil initiation nucleotide. Okay, it's always either A ataupun G. So kalau A dia kata 5 dekat sini. Okay. Alright, so dipanggil initiation nucleotide. So this is then based on this, dia barulah boleh berlakunya transcription. Kita nampak lah dekat sini adalah transcribe region. Okay, so it identify, okay, RNA polymerase dia identify this 95 region, then it binds. So this data box ni, panggil data box. Okay, this inner box helps to stabilize the complex. Then we have this initiation nucleotides, okay, before the process of transcription can occur. Boleh? Apa tadi Nadira?
Boleh doktor. So maksudnya positive one tu yang initiation nucleotide. Negative one?
Yep. Negative one, I think kalau saya lah, if saya pun dah sebenarnya ingat lupa sebenarnya sekejap both are initiation nucleotide, but dia tengok dia punya location. Kenapa dekat sini dipanggil minus and dekat plus? Okay, these are the promoter region. This is the region for promoter. Okay. So dekat sini adalah -1 adalah it tells you, okay, now next we are going to have this initiation nucleotide. So dia dah jadi +1 because it already located dalam transcribe region. Betul tak? Dia dekat in between transcriptional site dengan promoter. So dekat bahagian promoter region, it is indicated as a minus region. Betul tak? Okay, -35, we have this -10 and we have this -1. So dekat sini juga adalah ini, saya rasa adalah nucleotide, initiation nucleotide juga, but dia hanya membezakan plus minus ni adalah either dekat promoter region ataupun dekat transcribe region. That's it. Okay, Nadira?
Okay, doktor.
Alright, okay. Do we still have time? Action process ni panjang juga ni. Lagi 2 minit. You all ada kelas ke lepas ni?
Ada pukul 10.
Okay, so I think I'll stop here. So we continue next week. Saya akan habiskan by Monday, then we'll start the final topic by Wednesday lah kot. Okay? Ada soalan?
Doktor nak tanya pasal apa ni lab lagi. Doktor
ada
ada
mesej
kelas
saya tak?
lepas ni. Saya ada mesej.
Ya ke? Saya
ada.
pandang lah mesej awak. Nanti saya check balik lah mesej awak. Saya ada meeting at 10.
Okay.
Tapi tak apa, awak ada mesej kan? Saya cari. Mungkin saya terlepas pandang kot. Semalam
Semalam.
Okay, sekejap. Nanti saya ramai yang dah keluar ke apa? Saya nak.
Belum.
44 je today. Okay, alright. So, ada soalan lain?
Tak ada doktor.
Alright, so thank you very much. So, untuk group 1 hari ni, so I'll see you this afternoon at 2 o'clock. Sharp 2 petang. Okay, thank you very much semua. Terima kasih.
Thank you doktor.
Thank you,
Thank you doktor.
Thank you.